Welcome to part 2 of the cell cycle. So this one is all about regulation of the cell cycle, our last bullet here on our main themes for this chapter. And we can start by asking a question about evidence for cytoplasmic signals that might help control the cell cycle. And so we'll look at a couple of experiments here and with using mammalian cell culture. So here we see an example. You can grow cells in dishes like this. The cells grow on the bottom of the dish. You look at them with a microscope, and here we see these cells in culture. This is a real image of real cells, but we'll look at some cartoon versions of them. So in the experiment here, you can take cells at different stages of the cell cycle and fuse them. So cause the two cells to become one, and then see what happens as a consequence of that fusion. So here in experiment number 1, we're fusing two cells here. One is in S phase. Remember, that's synthesis phase. That's when DNA is being copied. And the one is in G1 phase. This is just before S phase. When we fuse them together, this second cell will enter S phase. So it's like it's getting something from the cytoplasm of this cell that it's fused with to say, oh, get into S phase. And it can do that. S phase would be the next phase anyway. But maybe you could argue, well, that maybe that cell was going to go into S phase any moment anyway. Let's look at experiment number 2. We're going to take now a cell at M phase. So this is during mitosis. And we're going to fuse it with a cell in G1. So this is before DNA has been copied. So what do we see here? Well, that cell that was in G1, it starts to act like it's in mitosis. Of course, it skipped over S phase, the synthesis phase. And so it's not just a matter of, well, that phase was next, because this certainly wouldn't have normally been next for this cell, but it started into mitosis anyway. So that tells us there's something in the cytoplasm of these cells that act as a signal to tell what to do next. What are those things? Well, they're proteins called cyclins, which is a convenient name. They help control the cell cycle, so they're called cyclins. And if you look at just the total amount of cyclins, there's a few different kinds, but just total cyclin levels at different times, where do we see them peak? From going from interphase into mitosis and drop back down. The cell's divided, so we're back to interphase, and then gearing up for another division. Cyclins increase, they drop back down, etc. So there's a few different kinds of cyclins. We see that example here, cyclin D, E, A, and B, named basically in order of their discovery. And you see different cyclins and these CDK protein complexes, just focus on the cyclin side of this, occurring at different points in the cell cycle. So these guys, D and E, are important early on here, going into S phase. And then as we get toward the end of G2, we see cyclin A decrease and cyclin B start to become dominant. So different cyclins are important at different stages. And these are working in concert with what we call cell cycle checkpoints. And so there are 3 checkpoints, and we'll go through each one. And these are kind of, you can think of them almost like questions. Is sort of everything okay? So there's a checkpoint here at G1 before S phase. There's a checkpoint all the way around here at G2 just before going into mitosis. And then there's this M or mitosis checkpoint that occurs during mitosis. All right, so without knowing too much about these checkpoints yet, what might happen if there were no checkpoints at all? All right, yeah, given the choices, all of those things are possible, right? A lot of potentially negative things could occur if there were no checkpoints. All right, so let's start with the G1 checkpoint. This is also known as the restriction endpoint. This is kind of asking that question of, should the cell start dividing or not? If the answer is no, that might be because the cell, this is a normal part of the cell function. It shunts off to this G0 phase or GO phase. I'll tell you about that in a second. Or it's because there's maybe some kind of damage to the cell. So if there's something wrong, like, okay, this cell shouldn't divide. Another outcome would be that the cell doesn't go to this GO phase, but the cell is actually marked for self-destruction. But if everything is looking good, then the cell will pass that checkpoint and enter into that S phase, that DNA replication phase. This is generally considered the most important checkpoint. Once we get past this, that's where we see these other checkpoints occur. We've already sort of passed the point where probably most of the damage could be done, say, during that DNA synthesis phase. All right, so some cells will shunt to this GO or G0 phase just as a normal part of their function. So cells aren't always dividing. Some, like say your skin cells, they're constantly being replaced. So those are regularly undergoing cell division. But some other cells that say differentiated cells, cardiac muscle, neurons, cells that form your bones, those are not actively dividing. Older cells, so senescent resting cells, those are not actively dividing. And so those might be shunted over here to GO. And again, going back to that whole checkpoint idea, another possibility of why a cell wouldn't pass this checkpoint is that there's something wrong. All right, the next checkpoint is the G2 checkpoint. So look at what's happened at this point. DNA has been synthesized, that's S phase, so everything's been copied, and the cell is grown and gearing up for entering into mitosis. And so kind of the question to ask here, the cell is quote unquote asking, is that DNA replication, did that occur correctly? And there are a whole set of different proteins and that can detect this damage and then set off a cascade of signals to say, stop this if there is a problem. So, and that will downregulate cyclins, which again, you need cyclins for this to continue. All right, the next phase, or next checkpoint, I should say, is that M checkpoint. This is sometimes called the spindle checkpoint. And this is really important. The question here is, are the chromosomes attached to the spindle? So we saw this during pro-metaphase. That's when that happens. Going into metaphase, those chromosomes need to be attached. If they're not, then when those things try to separate the sister chromatids, that isn't going to happen. So anaphase will not begin if those kinetochores are not attached. So once that occurs, then in comes that separase enzyme to cut those sister chromatids apart, and off it goes. All right, so think back. Progression through the M phase checkpoint is stimulated by a complex that does which of the following? All right, so here are the choices. There, A is the answer here. That separase enzyme cuts those cohesin proteins, separating those sister chromatids. Now you can continue into anaphase, and the cell can finish up mitosis. If we look a little more closely, a little bit of detail here. Here's pro-metaphase where we have an unattached sister chromatid. We see this top one here. What that's going to lead to is this bottom pathway here. The cyclin B will be degraded, and so that's going to stop the cell from going any further. Here we see mitotic exit. If, though, these chromatids are attached to their microtubules, then this securin is degraded, and that basically frees up separase to act, and it will cut those cohesins, and away we go. So looking at those stages of mitosis overlaid here with those checkpoints, once we pass that G2 checkpoint, we're into pro-metaphase, and that's sort of the test for, are the spindles attached? If everything is okay, then that M checkpoint is passed, and then anaphase can proceed. All right. A little bit of a different question. To stop a cell from ever synthesizing its DNA, what step could you block? All right, so given the choices there, it's the G1 checkpoint. That's the one that you'd want to be stopping the cell cycle always if you didn't want the cell to synthesize its DNA. The other two checkpoints, the horse is already out of the barn, DNA has already been replicated at that point. So it's only the G1 that you would be able to stop DNA replication. So what triggers a cell to grow? Well, one thing might be growth factors. So these are external signals that set off a signaling cascade and switch on expression of cyclins. So let's take the example of platelet-derived growth factor, PDGF. So this is important for platelet replication. Platelets are important in clotting, so you get a cut in your hand or wound healing. You need a lot of platelets to come in there and help that healing process. So they have to replicate. You need a bunch of them. And so they will release this growth factor that stimulates cell division. And so if we look a little more closely here, what's happening, here's this growth factor, here's a receptor on the cell surface, there's a signaling cascade, it amplifies the signal. And then as we see here, cyclins are expressed. So also are proteins important for S phase DNA replication, and off this cell goes and divides. And we can see here in this experiment how important a growth factor is. You can take this connective tissue sample, cut it up. This would be a simple way to start a cell culture. Put it on into a culture flask here, and without that growth factor, you see some cells, they're just kind of hanging out, they're not really growing. But when we add the growth factor, all of a sudden now the bottom of this flask becomes covered with cells, and they're happily dividing because they're getting that signal to divide, divide, divide. Okay, so most animal cells exhibit something called anchorage dependence. What do you think that means? Okay, right. So they have to be anchored to something. They need some sort of substrate to grow on. And that's evidenced by, like in this example, with a cell culture. So in a cell culture flask, the cells will grow on the bottom. And if we, in this showing here, we scrape some away, some will kind of come in and fill this gap here. Again, on the substrate here, on the surface. So that's the anchorage dependence. And the idea that they will come in and fill this space, this has to do with density-dependent inhibition. Notice the cells will come fill that space, but then really grow no more. They form what's called a monolayer here, and then they'll stop dividing once the space runs out. So this is density-dependent inhibition. Now, both of these things are important when we flip them on their head and think about the loss of control. So cancer cells have lost a control of that cell cycle. Maybe something's wrong with those checkpoints. Maybe there's some other problems. But cancer cells won't exhibit that density dependence inhibition or anchorage dependence. So here's a cancer cell culture. Notice they're just growing right over top of one another. We might see these cells floating around in the liquid media that we raise these cells on. So again, they don't need to be stuck to the bottom of this flask. That's that anchorage dependence. And so they've lost their ability to basically say, oh, don't divide, turn off. We don't need more of you. And that's really the problem with cancer and tumor cells and cancer is they've lost that ability to switch off. So cancer is just fundamentally a failure of the cell cycle. This is why in a lot of ways it's so difficult to deal with. I mean, they are your cells that have just lost their ability to, say, stop replicating. And then that might lead to other problems because they're just replicating unchecked, even if DNA damage has occurred, and so on. And there's two kinds of genes that are associated with these disruptions in the cell cycle. There's these tumor suppressor genes and proto-oncogenes. So let's start with tumor suppressor genes. Now, the name itself gives you a hint of what's happening here. In a normally functioning cell, we'll use this example of P53. So this is an important tumor suppressor gene. This is part of a pathway that detects DNA damage. So here, normal happy cell, DNA damage has occurred. Oh, P53 says, well, there's damage. Let's set off this pathway to repair it. Great, no problem. The DNA is repaired and the cells can divide. Or the DNA is not repaired and P53 is important for saying, this cell has got to go, and the cell will undergo this self-destructive pathway called apoptosis. Now, if P53 isn't working, you can start to imagine the problems that could occur. Here is DNA damage, but there's nobody there to check it. Nobody there to say this cell is a mistake and it should not be copied. And so those cells might divide. Maybe there's no consequence. Sometimes maybe that doesn't mean anything if it's localized. But if this is a rapidly dividing cell, or this cell is receiving growth factor signals, we'll get to that in a second, ultimately that might lead to a cell that just accumulates so many mutations that it's really not functioning correctly at all, and it's lost all control of the cell cycle. So you see damaged genes, maybe aberrant chromosome numbers. So let's look at that other set of genes, the proto-oncogenes. Now, onco refers to a tumor. So proto-oncogenes, these are genes that are sort of pre-cancerous, pre-tumor forming. And they're really just growth factor genes. So these are normal genes that are important for your cells to function typically. But they're proto-oncogenes because they have the potential that if something goes wrong with their regulation, where they're on all the time, like grow, 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 when they shouldn't be, that's when we would consider them oncogenes. They're stimulating that for those cells to constantly grow. And so usually it takes both these things to happen, where you've got a cell being signaled to grow and it's got some damage to its DNA checking mechanisms of those tumor suppressor genes, where we start to see tumor and cancer formation. And so cancer cells aren't really responding to the typical checkpoints. And so they're, again, incorporating errors. They may reach a point where they don't need any growth factors at all to grow. They're just being signaled to grow all the time by some other mechanism. They're making their own growth factor, or they don't need it at all, and that they've completely lost control of the cell cycle. Okay, so besides this uncontrollable division, what else might happen during tumor formation? Okay, so given your options there, it's that lack of cell death. So a cell that has errors incorporated into its genome should be told to not to divide, and even shunted into that self-destruction pathway, cell death. That's usually not happening. And so we can look at cancer cells, this progression. It's called transformation. Usually that's only used in cell culture, but the idea is the same even, say, in situ within an organism, in a living thing. So there's some kind of mutation that inactivates those tumor suppressor genes, right? So that checking mechanism. The cells are proliferating, maybe because they're receiving growth signals, and any of those errors that are already there, they're being amplified, and maybe new errors are incorporated. So the DNA repair genes are now not working. A mutation mutation turns on an oncogene, proto-oncogene. Now the cell's being told divide, divide, divide. So more damage happens. It accumulates over time, and that's what leads to cancer. So these cells also avoid immune detection, right? Maybe that's not too difficult, considering they are self. They're your own tissue. But sometimes they're caught by the immune system and destroyed. But it's obviously the ones that don't get caught that create the problems. So these cells might form what's called a benign tumor, a growth. Officially, this isn't cancer per se. It's just a tumor. But what's underlying the mechanism here is very similar. This usually is more easily dealt with than these other malignant tumors. So this could be surgically removed. And so the malignant tumors, this is where we're talking about actual cancer here. So these will spread and invade into surrounding tissues. They might break off and spread to distant tissues. That's called metastasis. And they'll form more tumors somewhere else. And obviously, these are much more difficult to deal with. And that is ultimately, technically, what cancer is this spread. Cancer, think of like astrological sign. The cancer is the crab. It's this idea that these tissues are sort of spreading out their tendrils, almost crab-like, within the tissue. So here's an example, that example from your textbook showing breast cancer, how it might spread. So starting as a small tumor here, here's why early detection might be so important with any kind of cancer. This potentially could still be removed easily enough. Now we're starting to see that malignancy. We see the cancer spreading, or the tumor spreading into surrounding tissues, and then in this case, ultimately metastasizing to some other location. So even if you were to catch the cancer in these later points, you might not be aware of where it's spread, right? And that's the problem. There might be tiny tumors all over the place at this stage. Not great. All right, so, well, probably some of you have had direct experience with maybe a family member who have had cancer or have personally experienced this. Those benign tumors are much easier to deal with. That doesn't mean it's a piece of cake, but they can be removed surgically. Radiation treatment is often done maybe together with that, so targeting those benign tumors. The malignant tumors, again, that's cancer, are much more difficult to deal with. And that's where you get into chemotherapy, because you don't know where they all are. Maybe you surgically remove what you can, and then chemotherapy is used to treat wherever those other cancer cells might be. So an example of a chemotherapeutic agent would be Taxol. And this is something that stops actively dividing cells. Why does that work? Because cancerous cells are very actively dividing. And so that will target those cancer cells, but of course that's stopping your own cells from dividing. So why do people experience hair loss and nausea? Because those are associated with cells that also have high turnover. And so those cells are not being able to actively divide like they normally would. And that's where you get that hair loss and nausea associated with chemotherapy. All right, so we'll talk some more about cancer in a later chapter, and that's it for this round of cell division.